So when we are looking to construct a polygenic risk score, it's important to have this background information and understanding of how populations are defined so that we don't let misclassification errors sort of pervade and then have downstream effects on our predictions in clinical applications of PRS. So when we are creating a polygenic risk score, this is the model that's used, and I'm sure you've seen this before, but for folks who don't have a mathematical background, maybe it's scary and you just sort of let it wash over you without really picking it apart. But I think it's important to understand that the goal of a polygenic risk score is to predict a phenotype in a target population. And in this case, it's an individual whose genotype data is going to be integrated into the model. That's this G here. And then these betas, or these weights that you are adding to, that you're multiplying by each genotype, and then summing all of those genotype SNP weight products across an entire genome for an individual, come from a GWAS that was done in a discovery population. So in that discovery population, you have SNPs that are coming up as statistically associated with the trait of interest, but typically those tag SNPs that are assessed and are coming up in these association studies are not the causal SNPs. Sometimes they can be, but most of the time they are just in linkage disequilibrium, or LD, with the causal SNP, and so that's why you're seeing the statistical association. So it just really relies on this LD structure that there are correlations in frequencies between the tag SNPs and the true causal SNPs for a particular trait. So you take these tag SNPs and you take their effect sizes that are generated through the GWAS, and you sum up all of the genotype SNP weight products, and you can do some adjustments to those weights based on different models that you're using, different population differences, what you know about those population differences. You might want to adjust the effect size. There are lots of fancy things that can be done, but this is the basics oversimplified construction of a polygenic risk score. And one of the two main reasons why this can be problematic, or why there can be issues with prediction in one population when the GWAS was done in a different population, is because of those global allele frequency differences and differences in linkage disequilibrium structure across the genomes. So if you sort of grossly categorize people into continental groups like African, European, and East Asian, which may not be that informative for certain applications, in this case, you do see sort of trends in minor allele frequencies up here in the top right. And across the panels here, C, D, and E, you can look at the sort of correlations between SNPs as the sort of visualization of the LD structure in the different ancestral populations. So there are differences in LD structure and in allele frequencies across populations that can have an impact on the portability of polygenic risk scores from one population to another. So what are the impacts of these differences? Well, I've tried to illustrate them here for you with a toy example of allele frequencies. So on the left, you have your discovery population with frequencies of the tag SNPs and causal SNPs being equal to one another because they're in LD. And that's by definition what that means. And in the target population, some of those allele frequencies are different based on differences in linkage disequilibrium or other factors like environmental effects, gene-environment interactions, and also gene-by-gene interactions. So this on the left here, you see that there's a breakdown between in the LD between the tag SNP and the causal SNP due to recombination that happened at some point in that population, such that those frequencies are no longer they're no longer linked to one another. Here in the middle, you still have this LD. They're close enough to each other that there isn't a recombination event, but there may be differential environmental effects. Maybe the environmental effect is much stronger on this trait in the target population versus the discovery population, which then changes the effect size. So, and you wouldn't necessarily be able to measure that. So if it's not being modeled, then you're sort of using the incorrect effect sizes. And we know that the heritability of a trait changes actually is reduced, the stronger an environmental effect is. So there's less genetic influence on trait variation in a population when there are very, very large environmental effects. And finally, you can have epistatic effects, gene by gene interactions between two causal SNPs, one of which may be an LD, but the other one which is not. And so that one you're not really able to capture, and this isn't really represented at all in a polygenic risk score. So there are lots of things that can factor in reality that can really impact the accuracy of a polygenic risk score. So there's been some great work on this. I credit Alicia Martin to really spearheading and doing a lot of this research to show us how prediction accuracy varies across populations, and that as the global genetic distance between discovery and target populations increases, the prediction accuracy of the risk score decreases. There is also some research to show how this can be improved, and generally speaking, that is using combined SNPs and weights from multiple discovery populations. That can improve the polygenic risk score prediction from all populations pretty well, but it will have the most impact in this case on an African population. So you'll do better for everybody if you use combined SNPs and weights from multiple populations, but you can do the best, where there's the greatest improvement, you could say, in the prediction accuracy for quote-unquote African populations. But we are limited, obviously, by our ability to do this in combining data from different populations by the lack of diversity in GWAS. We just, here on the right is a this is again from Alicia Martin, looking at the global population in billions of individuals across these sort of broad groupings of populations, so to speak, and then the fraction of those that are present in genome-wide association studies. And still today we see these very stark differences and huge overrepresentation of European individuals in GWAS, despite having a much lower percentage of the overall global population. So we need to have greater representation from these groups that are highly heterogeneous, genetically speaking. Europeans tend to be much less heterogeneous than say, half the world's population of Asia, and then when you add Africa to it, it just doesn't even compare. So we're really missing out on a lot of the genomic diversity that could be contributing to health and disease in global communities by just focusing on Europeans. And so this can lead to biased estimates of in prediction. Also, it can lead to false negative and false positive diagnoses in people with non-European ancestry, primarily. And ancestry is relative. We have to remember that everyone has African ancestry. If we go back about 200,000 years to before anatomically modern humans migrated out of the continent and populated the rest of the continent. So it's important to just keep in mind that when we talk about ancestry, putting somebody in one ancestry category is just not it's not a valid construct, and is increasingly less relevant as populations get more. So in summary, it's very common to have misclassification in our research and in clinical care, because the way that we define populations is just not valid in a lot of cases. And this is likely to become an increasingly big problem as each generation becomes more diverse. And it's really unclear what information is being used to group people together for the purposes of analyses, especially when you have research protocols that collect information from patients or participants that are based on the OMB census categories for race and ethnicity, and then they're reported in research results as, let's say, African ancestry, instead of actually reporting Black or African American. So we might think we're doing a better job by talking about ancestry rather than race in our research, so as to not reify these sort of racist assumptions or scientific racism. But unfortunately, by replacing the terms without actually changing the way that we fundamentally collect and categorize people, may just be burying the problem a little bit more deeply. And race, ethnicity, and ancestry are often conflated with one another. They're fluid and they're context specific. So we really can't rely on those as consistent categories to use moving forward. And it's important to note that scientific or biological racism is what maintains our racialized frameworks that we use in research and clinical care, and also use of those frameworks themselves may reinforce these underlying notions, conscious or unconscious. So when we talk about polygenic risk scores, they may have utility for some populations, mainly people of primarily European ancestry, depending on the context, right? So what's the condition? Is it a complex trait or not? What are the other diagnostic tools that we have available in clinical care, and how useful are they? A lot of studies have shown that PRS doesn't actually improve beyond the existing diagnostic capabilities. And also, what is the effect of the environment on the trait? Since we know that heritability or the genetic impact on trait variation in a population goes down with the stronger environmental effects. So we have to think about selection of what SNPs we're using and the weights that are assigned to those based on effect sizes, and understand that those depend on allele frequencies and other factors in the discovery and target populations in the construction of a polygenic risk score. And also we have to take into consideration the genomic background that will sort of globally reduce the accuracy between populations. So unfortunately, we're still limited by the lack of data from diverse populations, and until we have greater representation in genomics research, we won't even be able to assess what the full extent of health disparities could be as a result of genomics and precision medicine. So I urge us to proceed with caution, and I appreciate your time. I would like to acknowledge that this conference, the ACMG annual meeting of 2022, is being held on the traditional territory of the Cherokee, Shawnee, and Yuchi peoples. And you can learn more about this from a local anthropologist who's a professor at Vanderbilt in a 6-minute video. I created a tiny URL /ACMG22land. I'd also like to acknowledge NHGRI funding for ClinGen, of which I'm the co-chair of the Ancestry and Diversity Working Group, our PIs, as I mentioned, Alicia Martin for her research on polygenic risk scores in diverse populations, and Sonia Reed, who's our moderator and organizer of this session. And thanks to the organizers of ACMG for the invitation.